Branched-chain amino metabolism is the story of three essential amino acids: leucine, isoleucine, and valine. We will build an intuitive visual map of their transport, their first two degradative steps, key regulation, and why this pathway matters for physiology and disease. Here is the high-level map. First, branched-chain amino acids enter cells via L-type transporters. Next, cytosolic and mitochondrial branched-chain aminotransferases convert them to keto acids and make glutamate. Then, in mitochondria, the branched-chain keto acid dehydrogenase complex performs oxidative decarboxylation to acyl-CoA products. We will highlight the two enzymatic steps that we will focus on. Step 1. Aminotransferases. Cytosolic BC-ATC and mitochondrial BC-ATM are homodimers that use the cofactor PLP. They transfer the alpha amino group from the branched-chain amino acid to alpha-ketoglutarate, producing glutamate, while the carbon skeleton becomes the branched-chain keto acid, KIC from leucine, KAMV from isoleucine, and KIV from valine. Let us emphasize both isoenzymes. The products on this step are BCK as plus glutamate. Step 2. The branched-chain keto acid dehydrogenase complex in mitochondria. It has three enzyme components: E1, the decarboxylase; E2, the transacylase lipoyl core; and E3, the dihydrolipoamide dehydrogenase. Its activity is acutely controlled by phosphorylation. The kinase BCKDK inactivates, and the phosphatase PP2CM activates by dephosphorylation. Between organs, there is teamwork. In the rat, muscle shows strong aminotransferase activity and exports keto acids, while liver strongly oxidizes them. Humans differ. Human liver also has substantial aminotransferase, so it can oxidize in place. Here is a non-overlapping side-by-side summary. Leucine is more than fuel. It signals. In cells, leucine activates the RAG-GTP-asase, recruiting mTORC1 to the lysosome, where Reb turns it on. The effect is increased protein synthesis and reduced autophagy. In pancreatic beta cells, leucine also supports insulin release. A striking optimization is that mitochondrial BC-ATM can bind the E1 component of the dehydrogenase to form a metabolon. This docking channels the keto acid directly to E1 and speeds the reaction. Why does this pathway matter? With BCKDC defects, branched-chain amino acids and their keto acids accumulate, causing maple syrup urine disease. Because phosphorylation regulates flux, the kinase BCKDK and the phosphatase PP2CM determine the active fraction. And in cancer, BCAA catabolism, often via BCATC overexpression, feeds glutamate and biosynthesis to support growth. The key takeaways: 1. Branched-chain aminotransferases make the keto acids and glutamate. 2. The dehydrogenase complex, controlled by phosphorylation, is the rate-limiting step. 3. Leucine signals through mTORC1 to coordinate growth and autophagy. 4. Dysregulation links to maple syrup urine disease, neurodegeneration changes, and cancer metabolism. Thanks for learning with us. This visual map helps you see how branched-chain amino acid metabolism integrates transport, enzymology, regulation, and disease.